Welcome back. Let's look where we are in the course at the moment. Talked about the challenge of cancer and the causes, DNA damage. Talked about cancer as a disease. And in this part of the course, we're talking about the molecular biology of cancer. The last several lectures talks about viruses and how they bring oncogenes in, and those oncogenes can transform cells to a tumor. And that led to the discovery of proto-oncogenes inside uninfected cells, which is the majority of cancer, 90%. And those proto-oncogenes get mutated and activated in some way. And these are the gas pedals that stimulate cell reproduction. Now I want to talk about hereditary cancers and how hereditary cancers led to the discovery of a second class of important genes in cancer, the BRACEs, the tumor suppressor genes. My story for hereditary cancer begins with a quote from Louis Pasteur. Louis Pasteur once said, "Chance favors only the prepared mind." It's the story of how cancer patients led a physician-scientist to a major insight in the biology of cancer. The man's name is Alfred Knudsen. Knudsen graduated from Caltech, California Institute of Technology in Pasadena, with a bachelor's degree. And while there, he took genetics, science of heredity, from one of the major geneticists of the 20th century. He went on to get his MD degree at Columbia and a PhD back at Caltech. So he was an MD and a PhD, and ended up in a career as a clinician scientist. Early on in his career as a clinician, he saw a child with an abnormal eye. Now, normally when you take a picture, you see the pupil of the eye looks kind of reddish, right? This child instead had a white thing, the pupil of the eye. And it was almost diagnostic that there was a tumor at the back of the eye. The tumor is called a retinoblastoma, which is occurs in about one birth in 20,000 children. Retinoblastoma is a treatable cancer. Knudsen would use radiation to treat it. It's now laser therapy. And so people with retinoblastoma live a normal lifespan very often and have children, etc. Over the subsequent years, Knudsen saw more children with retinoblastoma. Now he noted that some of them had a single tumor, and most of them had, when they had a single tumor, had it only in one eye. About 75% of the kids had this. Others had several tumors. And when they had several tumors, usually they had them in both eyes. He was very curious about this. And so he looked at when these tumors arose. He reviewed the charts of all these children. And he plotted a graph of when did the tumor, these two different types of tumors, the single tumor in one eye and the multiple tumors in both eyes, when did they appear? And this was one of the most famous graphs in the annals of cancer research. Here's the graph. It comes directly from his research paper. And there is Knudsen pondering how to deal with this graph mathematically. We're plotting here the cases, and it's kind of like a Kaplan-Meier curve because we're plotting the cases not yet diagnosed. So we start with 1 and end up, of course, with 0 because all the cases are diagnosed. And the age in months. And if you look at the tumors in both eyes, they seem to happen sooner. And secondly, the tumors in both eyes followed a straight line. Okay, in other words, the incidence seemed to follow a straight line over time. Whereas if you look at the curve of the graph of the tumor in one eye, this is a logarithmic scale, the tumors in a single eye were slower to appear. They appeared at about age 4 or so, and they gradually more and more patients were noted. Knudsen noticed something else. The children who had tumors in both eyes almost always had a parent or sibling with retinoblastoma. So he said, oh, this might be hereditary. And the tumors with a single eye and a single tumor, those patients typically didn't have a relative. So he said, maybe those people are not hereditary, and the term for non-hereditary cancer is sporadic. So we've got hereditary cancer, inherited, sporadic, non-hereditary. So let's summarize. Hereditary cancer, hereditary retinoblastoma, tumors in both eyes. Early onset, multiple tumors. Kind of like every cell could give a tumor in the retina of the eye. Whereas sporadic, one eye, and typically later onset and single tumors. I said chance favors the prepared mind. Here is where Knudsen's PhD training came in. He knew about genetic mutations. He also knew that genetic mutations can occur in any cell, somatic cells all over the body, like the eye, or germline cells, the ones that give rise to the eggs and sperm. He also knew math. When he looked at these graphs, one curving slower than the other straight line, it reminded him of chemistry, which he had studied as both an undergraduate and a PhD student. Some chemical changes need only a single event to occur. So we're looking at two molecules in the solution, and they come together, and that's it. That's the chemical reaction. Single hit, it's called, single event. When you look at this event over time, because there's a whole lot of molecules in the solution, so there are thousands of molecules, and you plot how many of them have done it, it's a straight line over time. Single hit. Other chemical changes need two events to occur. So the molecules are going around, bumping randomly, and they bump in, and then they have to wait a little while, and something happens over here, and then the second event happens. Two events, intermediate stage. The graph here is slower, obviously, two events, and it's a curved line when you plot it. Knudsen saw that the pattern for hereditary retinoblastoma followed a one-event timing after birth, and the pattern for sporadic was two events happening after birth. And here were his insights. First, the key event in retinoblastoma were mutations. Second, the mutations, he surmised, were in a gene. Those gene product normally stops the cells of the retina from becoming a tumor. Obviously, a tumor suppressor. Third, that people, they do, he knew this from genetics, inherit two alleles or two genes for this gene, one from mother and one from father. And the forms of a gene are called alleles. So we get an allele from mom and an allele from dad. Fourth, both alleles must be mutated. Both breaks must be off to make the protein involved non-functional in the cell. So two hits have to happen. So Knudsen developed the two-hit hypothesis for cancer. In familial retinoblastoma, inherited retinoblastoma, the child is born with already one mutant allele that the child got from one of the parents. The other allele is normal, so the cell still is not cancerous. But over time, things happen, right? Mutations happen. And so after a certain period of time, the second mutation happened, and you have two alleles being mutant, and now you can get cancer. In sporadic retinoblastoma, both alleles are normal, and so you need two events to happen, two hits. The first hit to make one allele, second hit to make the second allele, and when you get both alleles mutated, then you can get cancer. Knudsen's hypothesis was groundbreaking, and it has been confirmed with lots of different types of cancer. The two-hit hypothesis is a major idea, and the idea of tumor suppressor genes is a major insight. This lecture, I'm going to describe how cancer in families can be inherited. I'm going to describe cancers that are inherited. I've just described one of them, retinoblastoma. And finally, I'm going to describe how susceptibility to cancer can be inherited. So first, the inheritance of cancer in families. Cancer in families happens with close relatives maybe 10 or 12% of the time. A lot of families have cancer in it, and it's not surprising in your own personal experience. But when you think about it, it shouldn't be that unusual because 1 person in 4 develops malignant cancer lifetime and 1 in 3 dies of it. So it's not surprising cancers run in families. Okay? I've met many people who say, well, there's a lot of breast cancer in our family. That's not surprising when you think about it because the ratio of lifetime of females getting cancer is about 1 in 10. So on a statistical basis, there might be a couple of people in a family. How do we determine if cancer in a family is hereditary or sporadic? One way to look at it is epidemiology. We can do cohort studies that have been done in Nordic countries. But here's like a case control study in Iceland. And I want to introduce Iceland to you as a laboratory of genetics. Iceland has a confined population. There's not many people moving in and out. It derives from a population that came in about 870 AD and settled this huge place. Many of the people are related. They have socialized medicine. There's a lot of data on health. A lot of people are related. A lot of families, a lot of genetics has been done. So a lot of genetic insights about cancer have come from studying the population of Iceland. When you do a case control study, you have a lung cancer patient, and you ask the question, what's the, and control who don't have lung cancer, what's the relative risk of another person in the same close relatives in the family having lung cancer? And you can correct for smoking and passive smoke, etc. You get a relative risk of about 2, which says that even correcting for environmental factors the best we can, there is a genetic component here going on, because one person has smoking and the other is more likely to get it. So from all this kind of information and the information I described from Knudsen studies, what are the criteria for hereditary cancer? We can look at 2 different criteria. First, for the family, more than 2 first-degree, that's not temperature, relatives with the same tumor. First-degree relatives are siblings and parents. More than 2 with the same rare tumor. For patients, remember retinoblastoma? Multiple tumors at an organ, which says all the cells have 1 mutation already, and we're just looking for a second. And second, young age at diagnosis. Remember the retinoblastoma. Overall, all the results of epidemiology and genetics indicate that about 5 to 7% of cancer is inherited as a single or a few genes, and maybe a couple more percent is inherited susceptibility to cancer. So maybe total close to 10%. Let's step back a minute. 10% cancer caused by viruses, 10% by heredity. That leaves 80% that's caused by something else, which is spontaneous and environmental mutation. In talking about hereditary cancer, I have to introduce the rules of inheritance, a review. For some people, for others, it's new. You remember Mendel and his pea plants, etc. This applies to humans as well. Human DNA, as we know, and genes are carried on human chromosomes. There they are. And you've seen this now. This is the 3rd time in the course. You see the chromosomes can be laid out in pairs. One chromosome comes from one parent, another from the other. And when a gamete or sex cell is produced, it's random which chromosome pair ends up in the gamete. So if you take one of my sperm, for example, and you make a survey, you say, is it my mom's chromosome 1 or my dad's? So it might be mother's chromosome 1, father's chromosome 2, mother's chromosome 3, etc. So you keep looking, and there's terrific variety. The rules of human heredity are the same as the rules of any genetics. Let me just follow through with one phenotype that's genetically determined, and that's earlobes. Some people have attached earlobes. I'm sure you're all tugging your ears now. And some people have free earlobes, and they look different. And there's a gene involved for this. So we can say, here's the heredity of it. So we can say, here's, and just to summarize here, this is a male, that's a female, and the light squares that are not filled with color are ones that have free earlobes, and the colored ones are attached earlobes. And here is a female, for example, with attached earlobes. There's a male with attached. Here's a male with free, etc. So this is a pedigree of humans. It's a couple of generations. So here's a woman, a child, and here's the father, and here's the grandfather. So we're looking at a family, a pedigree, and we'll see some of these as we do this lecture. Now, if we look at genes, alleles, right? Everybody's got 2 of them for the earlobe gene, which is true. We can abbreviate them F and F. Big F I'll stand for the free earlobes, and the little f for attached earlobes. Everybody's got 2 of them. So this person, this male, the granddad, has 2 Fs he got from his parents. But what does he have? Well, he's got free earlobes, so he must have one of the big Fs. And he obviously has a small f because 2 of his sons have attached earlobes, and they have 2 small f's. So we can determine genetics that way by looking at 2 alleles and how they're being transmitted. Okay, that's the end of earlobes. This is not an earlobe course. This is a course about cancer, so let's return to Knudsen's 2-hit hypothesis. When two alleles are identical, we call the situation homo, the same, zygous. The zygote is the fertilized egg. When the two are different, we call it heterozygous. Now let's look at the Knudsen thing for homo and heterozygous, because this is a very important term to keep in mind. In hereditary cancer, the children are born heterozygous. They have one allele that has the RB gene that's mutated and one allele that's normal. So they have a normal allele and a mutated allele. They're different, so they're heterozygous. After a mutation, they become homozygous for the mutated allele. So the medical geneticists call this a loss of heterozygosity. They don't call it a gain of homozygosity. They just call it a loss of heterozygosity. In sporadic, they are born homozygous normal because they have two normal alleles. The first mutation results in heterozygosity because they've got an abnormal and a normal allele. And the second mutation, loss of heterozygosity or gain of homozygosity, and they have two bad alleles, and then you get cancer. So with that introduction to families and cancer and genetics, now we can look at the cancers that are inherited and then susceptibility to cancer. So let me talk about several hereditary cancers. And the first one, obviously, is retinoblastoma. So I've said enough about that. Again, hereditary cancers are about 10% of the total. Most hereditary cancers begin in childhood because you've already got a bad gene. Now, the ideal model of retinoblastoma is that the sporadic form is the same as the hereditary form, and everybody with retinoblastoma has the same mutation of the same gene. But everything I've said in the course up to now says that that's probably not true for most cancers. Most cancers are caused by multiple genes. And so it's going to be a lot more complicated than inheritance of a single gene. That's probably why hereditary cancer per se is pretty rare. You might inherit a bad allele of a gene that might get cancer going, but remember, you might need a whole lot of other genes mutated for the cancer to become malignant. So let's move on to the second cancer on the list, which is colon cancer. Colon cancer begins as a small growth in the colon, the large intestine, as a polyp, and that's what you look for in a colonoscopy. The peak incidence is in the ages of the 60s. 98% of the colon cancer arises from polyps. There is a hereditary form of colon cancer. It's about 5%. 95% of colon cancer is sporadic. And in hereditary colon cancer, it's a phenomenal thing. Very often, I've showed you earlier on a picture of colon polyps, but here are just thousands of polyps in the colon of a teenager. I told you that most cancers happen late in life. Here's a teenager. And this teenager has a hereditary colon cancer. It's called FAP, familial adenomatous polyposis, FAP. But remember those letters FAP because I'll be coming back to the gene involved later on in the course. Multiple polyps early in life. Obviously, they inherit one bad gene, and early on in life, the second gene causes loss of heterozygosity or homozygous for the bad allele, and now they get cancer. It's a tumor suppressor gene. In stomach cancer, about 10% seems to run in families, but stomach cancer is mostly environmental. It's caused by H. pylori, Helicobacter pylori infection. There have been case control cohort studies of stomach cancer that show this. A very famous person who had stomach cancer was Napoleon Bonaparte. When Napoleon lay dying in 1821 at age 52 in St. Helena, one of the last things he exclaimed was, "Pylorus, my father's pylorus." It's the French word pylorus. Pylorus is part of the stomach. He was in incredible pain in his digestive system. His father died of stomach cancer. His grandfather died of stomach cancer. His brother, a brother died of stomach cancer. 3 sisters died of stomach cancer. Get the picture? And so did Napoleon, a famous person who died of stomach cancer. So he knew about hereditary stomach cancer as he lay dying. Third tumor, Wilms tumor of the kidney. It starts in the embryo of the kidney, and you see there's a tumor here at the end of the kidney. Here's the kidney, and here's a large 7-centimeter tumor in the kidney. These are blood vessels that are impinging the kidney. Here is a pattern of heredity, probably spontaneous mutation here in one of the parents. The Knudsen model applies here. If it's inherited, if this is an inherited kidney cancer, Wilms tumor, there will be bilateral, multiple tumors, early onset. If it's sporadic, there'll be a single tumor, right? One kidney, later onset. It's a fairly rare situation. It's very treatable now, chemotherapy, surgery, and radiation. And most people with Wilms tumor are now living to reproduce. Another hereditary cancer begins young usually is called neurofibromatosis. A neurofibroma is a tumor that follows the nerve tract. It's from the fibrous tissue in the nerve tracts of the body. Half it's hereditary, half is sporadic. It's pretty common. It's about 1 birth in 3,500. That's more common than some of the other cancers, which is about 1 in 20,000. There's a very high spontaneous mutation rate in the germline cells of parents to give rise to this. Its onset is typically in the first decade of life, and sometimes you can get skin lesions, other times eye lesions. Highly variable, sometimes little lesions, sometimes big ones. It can be a problem if tumors arise internally in the brain and blood. Fourth cancer, hereditary, breast cancer. If we do epidemiology of breast cancer and we decide that a family by those criteria has hereditary breast cancer, remember, multiple tumors, both breasts, early onset, more than one relative, blood relative with it. We can look in the family and see what the data show. So for example, here is a case control from 52 studies of 58,000 cases and 101,000 controls. If there's 0 relatives with breast cancer, the probability is defined as 1.0. It doesn't mean 1%, it just means 1.0. It's 10% lifetime. For 1 first-degree relative with it, it goes up to 1.8. For 2, it goes up to 3. And for 3 and above, it goes up. So the more it goes up to 4. So the more relatives who have breast cancer in the family, the more the risk is and the more probable it's hereditary. When you look at these families in terms of the risk of breast cancer, of getting breast cancer, it's an amazing set of information. At age 40, if a person has no mutant allele for breast cancer, if they're not homozygous for a bad allele by our assumptions of hereditary breast cancer, the chance of getting breast cancer is about 1 in 200. Okay? By age 50, it's about 1 in 50, etc. And you look down at the numbers and you see it's about 1 in 12 by age 70. If the person has 1 bad allele, that is, they've inherited from 1 parent who has breast cancer, and they've inherited 1 bad allele, that's enough to increase the ratio, the probability of 1 in 6. And by age 50, it's a ratio of 3 in 5. By age 60, it's 4 in 5. And by age 70, it's 5 in 6. An enormously increased risk if there is hereditary, breast cancer. The king of hereditary breast cancer, in a sense, is this family. It's an amazing family, founded by Drs. Lee and Frau Maney. And if you look at this family, here is a grandparent with breast cancer. Here's a daughter, and the 3 of them here with breast cancer. But they have multiple cancers in the family. It's not just hereditary breast cancer, it's multiple cancers. This implies maybe a single gene that's mutated that is affecting a number of different types of cells, and we'll see what it is later on. Inherited susceptibility to cancer can also occur. You know people that seem to be susceptible to various diseases. What about cancer? There are a number of genetically determined diseases that make people more susceptible to cancer, and these diseases involve DNA repair. You'll recall every day there are thousands of mutations that happen spontaneously to DNA, and if you can't repair those, you're going to be more susceptible to cancer. So here is a list of several of those syndromes. Here is DNA repair in the bottom. You see that one of them is called xeroderma pigmentosum, and we mentioned this before, they can't repair ultraviolet light, so they get skin cancer damage. The Bloom syndrome cannot unwind DNA for repair, and so they end up with lymphomas very often. And the Lynch syndrome, where they can't repair some DNA errors, ends up with colon cancer. What's the logic of this? Maybe their defect is in a repair gene that's only expressed in that particular type of cell. A consequence of this, the failure of repair of DNA damage, is in xeroderma pigmentosum, for example. In xeroderma pigmentosum, children are going to get skin cancer just by going outside because they can't repair ultraviolet light damage to DNA. So this graphic shows you the cumulative incidence in percent of a bunch of patients, and melanoma and other serious skin cancers are going to happen late in life typically, but they'll happen earlier in life with people with this particular disease. So that's inherited susceptibility for DNA repair. Here's another inherited susceptibility that brings back some chemistry we learned earlier on in the course. Remember where molecules get converted and activated in the liver? Remember that a molecule gets taken in by the body and it goes to the liver, like this one, and the molecule becomes more soluble. Remember that OH is put on to make it more soluble so it can be excreted. Now, the system that does this is a system that's inherited. It's a protein, actually. It's a series of proteins. It's a family of genes. And they have genetic variants like anything else. So you can envision that there might be genetic variants that don't do the activation really well. And indeed, that is the case. Here is a genetic variation in activation to form an active carcinogen. The active carcinogen in this case is benzpyrene. Benzpyrene, remember the active ingredient of cigarette smoke? That was a smoking gun for cancer we talked about before that bound to P53. It becomes activated in the liver and puts on these O's and stuff, you remember, and then it binds to G in DNA. And there are inherited variations in the ability of this activation event to occur. So people who have a genotype of inheriting two of the A alleles, it's not the basis in DNA, it's just a classification, have a very active gene and they're really good at taking cigarette smoke and activating it and getting cancer as a result. The people with the C allele do the conversion much less. So this is an explanation of something that all of us are familiar with. We all know smokers who are old and still smoking and don't get lung cancer. Maybe they got the right genes, the genes that don't activate this particular carcinogen. Later on, I'll describe the activation in this system for pharmaceuticals and drugs and how we can identify inherited variations in drug activation and inactivation to optimize therapy. The DNA sequencing that's going on of tumors now is a major effort, and we're looking for variants that might be associated with cancer susceptibility. So here's the idea. We have a sequence of DNA, and we look at a certain spot for a variation. So one person, most people might have an A and a T at a certain spot. And there's a whole bunch of people that have a G and a C and a G and a C and a G and a C. And you look at those people and you try to relate the genetic difference, that particular genetic difference, the GC replacing the normal AT, to cancer. And if that relates to cancer, then we can say that that might be part of the genes that are causing the cancer or making people susceptible to the cancer. So we can classify people having this change as common homozygotes, which are people who might be normal, and heterozygotes, so people have one gene that's bad, and variants, homozygotes. And we can classify those people, and the people who have the variant homozygotes might be the people most susceptible to cancer. This is an easy thing to do, to classify people on the basis of these small DNA variations, and you can begin then to say that these people might be more prone to getting certain types of cancer. In this lecture, I've introduced the concept of hereditary cancer. I described how cancer in families can be inherited and what the criteria are for defining a cancer as inherited. Then I described several cancers that we know are inherited. And then I described susceptibility genes, the genes that make people more prone to getting susceptible to getting cancer, and how those are inherited as well. In the next lectures, I'm going to look inside the cancer cell, see what the genes involved are, especially for those that are inherited, and what they do, with the hope that understanding those genes can lead to a treatment and a cure for this disease.